I'm a human being. Their professors are human beings, just like they are. And we're not, some evil mad scientists squirting stuff into tubes that don't like to interact with people, even though that might be true.
Right.
Not all of us.
Not ours.
Not
Right,
all.
exactly.
Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am David Peterson.
And I am Kim Klinowski, your host for today's episode. If you think rush hour traffic is bad, you should check out the endosomal cargo traffic.
Our guest today is Dr. Vincent Starai, faculty in the Department of Infectious Diseases in the Department of Microbiology and adjunct faculty in the Department of Cellular Biology. Vinny, welcome to the podcast.
Thanks a lot, everybody. Happy to be here.
We're happy to have you. So, Vinny, the first question that we like to ask our guests is about their pathway into science. So, can you tell us how you first became interested in science?
Sure. So for the vast majority of my adolescent life, I was really supposed to be a brain surgeon. So I think that's probably pretty typical of the people that you interview. We don't actually know when we're that young what science actually is and basic research. And so I did everything I was supposed to do to be competitive for medical school. When it came time to go to college, I thought I was going to be a pre-med major. It turns out I didn't know that pre-med isn't actually a major. I had to get a degree in something. So I scanned the list of different science-based degrees and I saw microbiology. And I thought, wait a minute, I'm going to be doing surgery under a microscope. So I'm going to go to University of Illinois to be a microbiologist so I can learn how to use a microscope. And so my first 2 years that I was there, I didn't take any microbiology classes. I didn't actually know what microbiology was other than it was like small things. And I wasn't a particularly good student. Academically, I was fine, but I liked the party scene and I liked spending a lot of time outside of the classroom. And I realized that I wasn't spending enough time doing all of these things that we tell our students that we have to do to be competitive for medical school, clinical shadowing and research and all these things. And so I realized, you know what? I've got to start doing something. And of course, I was at a party and I met completely by accident a research technician in a microbiology lab. And I asked him, hey, I'm a microbiology major. I kind of want to start learning what happens in a laboratory. And he's like, yeah, come on in. So they hired me as a dishwasher. So I spent a semester washing dishes, making media, not really caring what was kind of happening in the laboratory, because in my mind, I'm thinking, hey, I'm checking this box. I'm going to get this experience, even though I'm only washing dishes. And this is going to make me competitive for medical school. Clearly, I had no idea what I was doing. And after that semester ended, I went to my PI. As you are fully aware, PIs, so professors, are some of the most scary people in the world, right, to interact with and to talk to. So I worked up all my nerve and went to the PI of the laboratory, John Cronin at Illinois. And I asked him, hey, can I join your laboratory as an undergraduate to do like an independent project? He's like, yeah, sure, whatever. And so that next semester, I started working in the laboratory, again, not really paying attention to what I was doing, just learning how to streak bacteria on a plate, learn how to do transformations and so on and so forth. And eventually I was given a project where I had to identify a mutant derivative of E. coli that couldn't make lipoic acid. Okay, so it's an essential cofactor. And I picked and patched probably 13,000 colonies or something over that summer period. While my friends were out having fun back homes for the summer, I'm sitting in a laboratory using, 13,000 wooden sticks. And the vast majority, of course, of cells I was looking at were not the right phenotype, right? They just grew like normal cells. And then I found one, right? I found one that didn't grow if we didn't provide lipoic acid. I was like, wait a minute. That's kind of cool. I think I just learned something important about biology from this little stripe of smelly goo. Just because it didn't grow here, now I'm understanding how genes are important and they encode for proteins, and these proteins have an enzymatic activity and that helps them sort of live. And that was a very striking moment. And it was that point that I realized, wait a minute, I think I really like this research stuff. So then I actually started paying attention to what I was doing, and not only what I was doing, what the students around me were doing, what the faculty members were doing. I started actually paying attention to what was happening in the classroom, even though my grades were fine. And in my, I think in my last semester of my undergraduate career, I realized I was taking a class by the PI that was actually in my running my laboratory, and watched him use like props to kind of explain how DNA gyrase worked. And I was like, that's really cool. I like this whole teaching thing too. I think I might want to be a research professor at a university. And sure enough, that's where I am now.
So that's great. So you went from lipoic acid oxytrophs, is that the correct term?
You got
Wow.
it. Yeah.
I still remember some of my micro.
Yeah, I like it.
To using yeast as a model organism for another organism that is an endosymbiont of a parasitic worm.
Right. All right. Do you want me to get you there?
Yeah.
Please do. Let's get you there. Okay. So in my graduate education at the University of Wisconsin, I stayed in microbiology. I did short chain fatty acid catabolism and salmonella, so basically how bacteria eat lipids or fats for energy. And it was a great experience. I thought I would stay researching bacteria my whole life. And I realized as I was getting close to the end of my PhD that I wanted to learn something a little bit different. At that point, I was doing a lot of pure protein biochemistry in the laboratory. I liked that. And I was like, I think I want to keep doing this, but I don't know if I want to learn something different or just stay doing the same stuff. And eventually, I went to Dartmouth Medical School for my postdoctoral research experience, and I decided to move into a eukaryotic based system. And so this is where I learned basically the cellular biology of yeast. So in my postdoctoral experience, I explored the biochemical regulation of intracellular membrane fusion in eukaryotes. So learning how transport vesicles contact their target organelles inside the cell, interact with these two lipid vesicles, and fuse together to deliver cargo. And I really enjoyed that. So I'm learning now a brand new system, how to manipulate a eukaryote as opposed to a bacterium. As techniques were very similar, so the switch was relatively easy. But now I'm sort of diversifying my own research foundation. So learning classical techniques both in a eukaryote and in a bacterium. And as I become more and more familiar with the yeast-based system, I realize, hey, this organism is a really good model for a typical eukaryotic cell. A lot of those central pathways are conserved across all eukaryotic cells. Yeah, they get more fancy, I think, as you get a little bit more advanced in the cell type. But for the core pathways, it's actually pretty good. So when it came time to start my own lab, go out on the job market looking for a university that I could trick into giving me a job, I decided that I wanted to combine sort of both of my research paths and utilize yeast as a classical eukaryotic cell model system for the study of intracellular bacteria, which are known to secrete proteins into their host cells to rearrange that eukaryotic host cell in order to survive, to prevent their degradation, to rebuild membranes around the bacterium so that they could persist, so that they can eventually cause disease. They don't really want to cause disease. They just do it as a side effect of just trying to live. And yeah, so I said, okay, now I can use this yeast-based system to understand membrane traffic at a very high level. So I'm going to identify these bacterial proteins that maybe impact membrane traffic, because being able to manipulate membrane traffic, or the ability of the cell to move proteins and membranes around the cell during development or during growth, it's a very similar process again from yeast to man. So I think I can do this. And it allowed me to do what I tell other people I do: host pathogen interactions without a host and without a pathogen, right? So we take these proteins from bacteria that we're pretty sure cause or are important for their ability of the bacterium to survive in their eukaryotic cell host, and we express them in yeast, and we look for them to do something bad, whether it kills the yeast, whether it disrupts intracellular organelle morphology, disrupts the cytoskeleton. And then we can use sort of the power of yeast genetics and biochemistry to really characterize these proteins and try to figure out what they do. And so my most recent research that that we're going to talk about today is the idea that this bacterium, this Wolbachia bacterium, lives inside of a filarial nematode. This is a little worm that actually can cause pretty severe disease in humans. And that relationship is absolutely essential. The bacterium must be inside this nematode for the nematode to not only survive and reproduce, but also to cause disease in humans. The big problem is you can't really grow either of those organisms well. You can't grow Wolbachia at all. We have no genetic tools for the organism. And the worm we can grow, but there are no real genetic tools. We can't make mutations, so we can't really explore this biological system with any of our current technologies. So this is sort of the perfect system or the perfect problem to address with my yeast-based system that we can grow very easily in the laboratory and use that yeast as a representative of the nematode or the worm cells that the bacterium survives inside of.
So how do you pick which Wolbachia proteins you want to focus on?
Sure. So the bacteria in general, those that live inside of the cells, they often produce proteins that get secreted into that eukaryote to sort of modulate that cellular biology of the host. And a lot of times those proteins look pretty similar to each other across the bacteria because they all are targeting very similar pathways. So we can look at the genome, the genomic sequence of this bacterium, pick out the proteins that might look like those secreted proteins and pick those. We can also take a look at the Wolbachia bacterium and look for genes that encode for proteins that actually look more eukaryotic like. So that these proteins are probably mimicking or hijacking the normal eukaryotic pathway to benefit the bacterium. And so proteins that look more eukaryotic, we pick proteins that look like other secreted proteins from other intracellular bacteria, we pick. And we also pick proteins that other researchers have identified in basically squished worms. So they take the worms and harvest them, and they identify proteins from Wolbachia that are actually in the tissues. And these are proteins that are likely released from the bacterium during that endosymbiosis.
So, just to be clear for our listeners, the Wolbachia is the bacteria that's not necessarily causing the disease. It's the
Correct.
worm
That's
that's
right.
causing disease. So, why do we care about the Wolbachia and why are we studying?
Right. So we care about the Wolbachia because we know that if we can figure out how to prevent Wolbachia's ability to persist inside of the nematode, then we can also prevent the nematode from being pathogenic to humans. And even in animals, since the worms that we study, Brugia malayi, which is a human pathogen, there is a very close relative, Dirophilaria immitis, which is canine heartworm. Canine heartworm
Oh,
also requires Wolbachia for survival and to cause disease. The bacterium itself is not the pathogen, but it allows the worm to be the thing that makes somebody or something sick.
so can the worm exist without the bacteria?
The worm can exist without the bacterium, but it pretty much hates its life. It turns out the bacterium is a reproductive pathogen or parasite. So it actually invades the reproductive system of these nematodes. And in the absence of the Wolbachia bacterium, the nematodes are completely unable to reproduce.
So they're sterile.
So they're essentially sterile. Yeah.
Interesting. So is the Wolbachia and the two worms you mentioned, so the Brugia and the Dirophilaria, is it the same species of Wolbachia or close
Yeah.
or?
Yeah. So Wolbachia, all of the different Wolbachia out there, actually, I should note that probably up to 70% of the world's insects and arthropods can actually be considered hosts for Wolbachia because there are actually versions of Wolbachia that persist quite well in flies and mosquitoes and so on and so forth. And those Wolbachia are actually a little bit different than the Wolbachia that persist inside these filarial nematodes. But as of right now, they're all the same genus and species, Wolbachia pipientis. And there has been some, a couple of decades ago, there was kind of a movement to try to respeciate Wolbachia because they actually do very different things and they live in very different hosts. But right now, everybody's the same, Wolbachia pipientis. But at the genomic level, they definitely have differences, especially in the classes of those proteins that get secreted from the bacterium into the host.
Because those have evolved to be specifically fit to interact with their host
That's
proteins.
right. That's right. Because the Wolbachia in like insects and arthropods, those Wolbachia are not essential for the reproduction of those other insects for the most part. And so it's a much more parasitic interaction with those fruit flies and mosquitoes and so on and so forth. And because those Wolbachia effector or secreted proteins are very different between those populations, worm Wolbachia cannot live in flies and fly Wolbachias cannot live in worms.
So in probably high school biology class, we learn about the mitochondria, the powerhouse of the cell,
That is true.
chloroplasts.
Yep.
So where is Wolbachia on the path of invading bacteria to established organelles?
Right. Actually, that's an interesting question. So, we have very strong evidence that organelles like the mitochondria and some other parasites, the apicoplast, these are all at one point bacteria that were captured by some other cell. And eventually we evolved to maintain this very tight relationship, an essential relationship between the mitochondrion and that host cell, which eventually became that eukaryotic cell. And we feel very strongly that the mitochondrion probably evolved from some ancient alpha proteobacteria. So it's just a class of bacteria. And it turns out that Wolbachia in itself is an alpha proteobacteria and does have some similarities to maybe what the mitochondrion can do. And in fact, in Brugia and in flies and in mosquitoes, the Wolbachia in females actually invades the germline. So it invades the ovaries and is maintained in that germline at sexual reproduction so that all offspring actually get their Wolbachia from their mothers. So much like mitochondria, Wolbachia is maternally transmitted to offspring, which allows Wolbachia to stay in that population essentially until somebody tries to get rid of it with antibiotics.
That's fascinating.
Yeah.
Oh man, Wolbachia does some crazy stuff to like flies, for example. It can feminize male flies. Does this thing called cytoplasmic incompatibility to ensure that only female flies can actually become or have viable offspring. So an infected male fly that's infected with Wolbachia, if it tries to mate with an uninfected female, none of those offspring are viable because Wolbachia is trying to be selfish and keep itself in that population. Yeah, it's crazy.
Very smart organism.
That is actually far smarter than we are.
Actually kind of scary.
It's terrifying. Yeah, absolutely. Yeah.
So I've also read that Wolbachia is being used in some public health programs.
Fact.
So can we talk about that, the flip side?
Sure. Yeah, so Wolbachia, when it associates with and infects mosquitoes, for example, the mosquito does actually mount a response to the presence of that bacterium. And it's thought that the response to that bacterium actually it actually either suppresses the immune system or it causes other changes in that insect to make it actually a less capable carrier of other diseases, of other viruses, for example. And I believe there have been some sort of medium scale studies, I believe in Australia, where they've released Wolbachia infected mosquitoes into the environment to try to create a population of mosquitoes that are worse carriers of dengue. And this has actually been effective, where now the dengue load in these environments has been strongly reduced because of the Wolbachia presence in this population of mosquitoes, which normally didn't carry Wolbachia. And because of how sneaky Wolbachia is to maintain its presence in this insect population, it very rapidly spreads to the insect or to the mosquito population, so that all of these mosquitoes now basically suck at carrying dengue.
That is fascinating. So apparently the Wolbachia doesn't have much of a negative effect on the mosquitoes?
Correct. There is some changes in their reproductive capacity, but the mosquito themselves, they don't care if they have Wolbachia. They do pretty well with or without the Wolbachia.
Wow.
So, Vinny, in addition to research, you have a particular affinity for teaching.
I do.
Can you tell us why teaching is so important to you?
Yeah, so that's actually a great question. So, as you guys know and understand, when we're being trained as research scientists, as a graduate student, and then eventually as a postdoc, and then come into our own laboratories, we're never actually taught how to teach, right? And I know there might be a bunch of undergraduates out there listening to this, and they say, "Oh yeah, I have these faculty in my classroom," right? They're not taught how to teach. They don't really want to teach. I spent 12 years learning how to squirt things into tubes, these small volumes of colorless liquids back and forth, right? And my only experiences of teaching were as a teaching assistant, as a graduate student. And usually there it's don't stick your fingers in the gel box while it's running, right? And make sure you balance your centrifuges. And so when I started teaching, so when I started at UGA, I was given 18 months of no teaching time to set up my laboratory and to begin my research program. About at the 12-month mark, my department head came to me and said, "Okay, this is the class you're going to teach. We haven't taught this class in about a decade, so we don't have any material for it. Go." Right? And so I spent an entire summer trying to figure out what I was going to teach in this sort of upper level microbial biology class and put together my slides. I show up on the first day of class. I have about 80 students looking down at me, waiting for me to say something. And I'm thinking to myself, what the hell am I doing here? Right. And then because I have to, you start the process and you go on and you do you go through your lectures to the best of your abilities. You write the exams. There's an art to writing an exam. I had no idea that you have to learn how to write an exam.
That's a tough thing to
Yeah.
learn.
Oh, absolutely. I'm still learning how to do it. And by the end of the semester, I was getting a lot of sort of student interactions, just sort of talking about the class, talking about their lives, talking about their assignments in other classes. And I thought to myself, wait a minute, I'm actually sort of reaching out and interacting with these students and providing them information that I never got as an undergraduate. I didn't know where to look for any of this. Like, I didn't even know that research was a thing for an undergraduate until I was probably like in my third year of college. And that really kind of struck me. I was like, wait a minute, I can actually make an impact on these particular students. And I took a look at my classroom end of semester evaluations from students, and they gave me actually pretty good scores, which was surprising. And I'm like, wait a minute, I think I'm actually good at this. I have the ability to transmit information, despite the struggles of learning how to teach a class. And so from that point on, when I realized that I can actually really give these students a good information, give them the knowledge that they didn't know where to find the information to. So where can I go for internships, and what does application to graduate school look like, and so on and so forth. I made a very strong commitment to myself to really be that faculty member that actively transmits this information to students during class and outside of class. So now teaching is a very important part of what I do on campus here.
Yeah, I think I absolutely agree with you about the fact that we are not trained as scientists as we're maturing as scientists how to teach.
Right.
And we arrive here and all of a sudden we've got to teach and we're trying to remember professors that were good and ones that were
Right.
bad. And
Why? Yep.
we I do have to give a shout out to the Center for Teaching and Learning here at UGA, the CTL, because they have some nice workshops and I've participated in these when I have
Sure.
time, but time is
Yeah.
precious.
Time is precious, but it's up to us to decide how we want to split our time and then be okay with that,
Hmm.
you know? So because I put more effort into teaching and educating these students, I put a little bit less effort into squirting things into tubes and so on and so forth. So, there's the balance that we have to make as we do this job. And as you all know, the recent years, our workloads as sort of education has changed a little bit. Our workloads are increasing, and it's becoming more and more difficult to find all of the time that we need to do all of the things. So, we have to be happy with the things that we choose. And right now, I love what I'm doing in terms of teaching as well as in research.
And I do think that that's changing. I think, as someone who reviews these transitional grants for people early in their career, I can see a lot more people adding training that they're getting at their home institutions and teaching,
Yeah.
managing labs,
Yeah.
managing budgets, all those things that we didn't have. I hate to say that we were walking uphill in the snow without shoes on when it was our time, but
That's exactly right, though.
it was a big struggle and a big transition. I mean, I remember being asked to submit my teaching plan, and I'm like, I wouldn't
Right.
even know people plan for teaching, right?
Right. They have where you have to, when we apply for jobs, you have to give something like a teaching philosophy. Right?
Right.
What is a teaching philosophy?
Right.
You've never taught. I don't have, my philosophy is I say something, you learn it, you get an A on the exam, right? So I understand what that is now. I've been involved in events with the Center for Teaching and Learning. I've been awarded for my teaching at UGA. And, I think that it's a difficult thing to really be passionate about the teaching, forget about your research, or kind of make that research a little less important. But it's so important to this new generation of potential scientists, but not just scientists. These are going to be our doctors, our dentists, our lawyers, and so on and so forth. So, it's a critical part of our mission.
And I do want to touch on something you mentioned. You were recently awarded the Sandy Beaver Excellence in Teaching Award.
I
So
was.
congratulations
Thank you
on
so
that.
much. Thank you so much.
So, Vinny, what do you think makes a good teacher, and how, as scientists, can we be better teachers?
Ah,
Right now.
That's actually a good question. I don't know. A part of being a good teacher, I think, is having that personality, right?
And
Like,
Vinny has absolutely no
no,
personality.
I have no personality. And so, one of the problems to be a really good scientist, in my mind, we all have to have a certain amount of social interaction defects, right? And we do, right? We would much rather be in the lab hugging our yeast or whatever than interacting with people. And so at some point, I think the ability to be a good teacher is somewhat inherent, or you have to kind of get over it with continued, practice. And it is something that you can practice, right? It is not, while I think a lot of our characteristics that makes us a good teacher are somewhat inherent, you can actually become better at it. And I think the biggest thing for me that got me really interested or really passionate about teaching and learning how to be a better teacher is simply talking to my students. What are the things that are working in the classroom? So not last semester, but the semester before in one of my classes that I've been teaching the same class for quite a long time here. At the end of the semester, I always ask the students for extra credit, actually, what can I do better in this class so that it's better for you or that you learn more? And based on those answers that I get at the end of the semester, I actually try to make modifications to the course in subsequent semesters. So, for example, this past semester, based on previous comments I got, I started doing group exams. At least a portion of the exam was done in a group format. I actually don't like group work, especially at the upper levels. I don't think it's very effective. But allowing the students to see 10% of the exam before they take the exam individually, and giving them the opportunity to talk with their classmates about the answers and things like that has really helped. So my averages in the class went up. It's already kind of a difficult class, but my averages went up. The students unanimously loved this, even though they had to take the bulk of the exam by themselves. They were able to actually work with their peers to kind of work through the questions, and they all did very well at that. I've made modifications to the material for things that the students like or don't like. And so listening to the students' feedback and then making those changes has proven to be quite successful for me in being that good teacher, right? And then to be able to relay my experiences to my colleagues and say, hey, you know what really worked? This worked. And or this didn't work. Yeah, it's not going to work for everybody. But yeah, I found that simply talking to my students, and not just talking to my students about the class, talking to them about their lives, letting them know that I'm a human being. Their professors are human beings, just like they are. And we're not, some evil mad scientists squirting stuff into tubes that don't like to interact with people, even though that might be true.
Right.
Not all of us.
Not ours.
Not
Right,
all.
exactly.
But I do want to say what you're making is just a really good point because, and it should be simpler in some ways because you're asking people to be scientists in the classroom.
Yes.
The classroom is now the laboratory, right?
Exactly.
So you have a hypothesis or your lesson plan, right? You're laying out your lesson plan, you're collecting data, which is the student's feedback, you're analyzing the data and deciding if you want to revise your hypothesis. And then you're disseminating your information to other faculty members. So you're technically just being a
I
scientist
technically am being a scientist. I
in the
suppose.
classroom
I
laboratory.
simply cannot get away from it. Yeah, fortunately.
Yes. No, but I mean that just means that we all have the potential of scientists.
We
to be
all,
teachers.
yeah, theoretically, that is true.
Absolutely. So moving away from teaching or research,
Lovely.
how do you spend your abundant free
My
time?
abundant free time? Well, so we're trying to figure out what free time actually means. So I have to sort of preface this with my life is extremely chaotic and perhaps the best way possible. So when I go home at night, I go home to 4 kids, a wife, 8 cats, 2 dogs, 2 large fish tanks, a leopard gecko, 3 tarantulas, 8 chickens, and so on and so forth. So my house is, and it's not that big of a house at all. So it's just like we live in this constant milieu of living organisms, right?
I'm hoping the chickens are out back.
The chickens are, in fact, out back, although that didn't help much. I think 2 summers ago, we had 22 cats in our house at one time. And yeah, we foster, and it's a lot of fun. So I come home and it's chaos, right? The house is a mess, and the kids are running around, and it's absolutely wonderful. But, as a child of the 80s, I grew up with video games, having that first 8-bit NES in the house. I still, even though I'm 48 now, I still like to spend some time playing video games. I watch anime. I try to do the things that bring me joy because, science has a way of kind of ripping out your humanity, right? It really does, because you've really got to work hard. You got to get that paper out before you get scooped, or you need this for that next grant. And you're going to do that at the expense of everything else in your life. And, maybe about 5 or 6 years ago, 7 years ago, I really decided to change. I can't live like this anymore. Maybe it was the Zoloft, finally getting Zoloft. But, I I disengage from science for the most part. My job, at 5:30, I go home, I spend time with the family. I do the things that I like to do. I do spend the time with my wife to remember who we are as, or were as a couple, getting together. That's something that we lose sight of after being together for, 21, 25 years. And we sit and we watch silly YouTubes. And yeah, just remembering to do the things that make us us, right? And that for me is my family.
Okay. Well, our guest today is Dr. Vincent Starai, faculty in the Department of Infectious Diseases and in Microbiology and adjunct in Cellular Biology. Vinny, thanks so much for being with us.
Thank you so much, David. Thanks, Kim.
So links to additional information can be found on our Instagram account, which is at PPP Podcast UGA.
As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes launched this season. Please rate the podcast. And if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Special thanks to our podcast assistant, Sid Wiggin, for research, editing, scheduling, and keeping us on track.